Hi, thank you very much for having me here today for what looks like and has already proven to be a very enriching couple of days. So you know better than me that protein function or misfunction can rely very much on the physical state that a protein inhabits. And protein can range from being diffuse to liquid demixed to form hydrogels or insoluble aggregates such as amyloids. And mutations can obviously shift you left and right in this spectrum with consequences for the cellular phenotype. The simplest example you can think of is a protein that is supposed to perform its function in the soluble state, and upon a destabilizing mutation, it starts to precipitate and aggregate and it loses function. But for many proteins, loss of function is not able to explain completely the phenotypes that you see upon mutation. And this is because there are a number of gain of toxic function mechanisms taking place induced by a mutation and forcing the protein to transition into a non-functional state, which is physically distinct from the functional one. A good example of these proteins that acquire gain of toxic function upon mutation is given by disordered prion-like domains. These are extremely disordered large regions in the human proteome that are really low in sequence complexity with very repetitive sequences as well. And they're particularly enriched in proteins that are able to bind nucleic acids, both DNA or RNA. But in vivo and in vitro, they can form these very nice liquid demixed droplets. They form insoluble aggregates. And the reason I'm interested in them is because they have become clearly implicated as genetic factors in a number of neurodegenerative conditions. But the question here really is like, does one of these different physical states explain the toxicity that you see upon mutation? And we started to think about addressing this by deep mutagenesis. And of course, the first thing we wanted to do was to have quantitative and accurate readout of cellular toxicity. And as a model, we took the TDP-43 C-terminal domain, which is a prion-like domain. It's a large disorder regions around 140 amino acids. And we chose this protein because, well, aggregates of TDP-43 are found actually in most cases of amyotrophic lateral sclerosis in the motor neurons of patients that are analyzed post-mortem. And TDP-43 is also, well, mutations in TDP-43 are also linked to familiar forms of ALS with pathological mutations clustering inside the prion-like domain. And what we did is a very simple selection assay, it's a bit similar to what we've just seen for alpha-synuclein. So we expressed TDP-43 with like a mutated region inside yeast cell. We sequence before protein expression or after 5 or 6 generations of protein expression. And we quantify toxicity by comparing the frequency of each variant in the output over the input and in a way that is relative to wild type. The libraries that we generated actually include all single amino acid substitutions, but also a lot of double amino acid substitutions. And if we focus on the single amino acid changes, this is how the data looks like. This is a heat map of toxicity for a portion of almost 90 residues inside the disorder domain with red stuff, meaning stuff is more toxic. It's increasing toxicity. This mutation is increasing toxicity with respect to wild type TDP-43, and the purple ones are decreasing toxicity. So what is really already evident by this heat map is that there's a hotspot of toxicity where the effect of mutation is really more extreme than anywhere else. Once you really enter the more low complexity region, if you notice, the hotspot corresponds to an area with a few more hydrophobic amino acids. Once you move to the side of it, where the sequence is really low complexity, the effects are really quite moderate. And the other obvious thing that you see, besides the hotspot, is that there's a massive effect of the type of amino acid that you introduce there, with hydrophobic amino acids leading to a decrease in toxicity, and instead polar or charged amino acids increasing toxicity substantially. And so another interesting and key factor I would say about this data set is that all ALS mutations moderately increase toxicity. And that's nice because in yeast, you do see stuff that is way more toxic, but these mutations would probably be embryonic lethal in humans. So I was quite happy to see that these mutations are just slightly off from wild type. because that's normally when you have disease. Since we had such an effect of amino acid type, we decided to analyze this in a more systematic way. So first of all, we took a long list of physicochemical properties and physicochemical scales describing amino acids, and we reduced their redundancy by PCA, and then we looked at which one of this scale was best at predicting toxicity given by mutations in this region. And to our surprise, you can explain basically almost 80% of the variance in toxicity on the basis of one unique property, which is our PC1, that is basically hydrophobicity. And even more to our surprise, because when we set out this project, we thought, okay, the more hydrophobic, the more aggregation prone, and the more toxic. But actually, hydrophobicity completely anti-correlates with toxicity. So the more hydrophobic, probably the more aggregation prone, but the least toxic. We also tested a bunch of other predictors and scales, but hydrophobicity alone is really the best scoring one. And if you look at aggregation propensity after correcting for hydrophobicity, you can explain only roughly 4% more of the variance in toxicity. And again, increasing hydrophobicity and increasing aggregation actually decreases toxicity. We wanted to believe this by looking at what was happening inside the cell. And we started looking at a bunch of toxic versus non-toxic variants that you see here tagged in green, and in red you have the yeast nucleus. And what we realized quite quickly was that the toxic variants were forming like spherical aggregates around the nuclear periphery, while the non-toxic ones were rather forming one or more large cytoplasmic aggregates. But the main difference between these types of foci came when we characterized them by recovery after photobleaching or other biophysical strategies, and realized that the blobs or the foci around the nuclear periphery were really liquid-like and recovering really fast after photobleaching, while the ones in the cytoplasm corresponding to the non-toxic mutations were actually rock solid. So hinting at the fact that probably increasing the hydrophobicity of the sequence is just causing a very efficient aggregation process, and that this is somehow beneficial for the yeast cell at least. You may wonder why we also designed the libraries to have so many double mutants. And the reason is that having a lot of double mutants inside our selection experiments allowed us to start working in the direction of understanding if this disorder domain is really disordered inside the cell. In the phase separation field, there's a massive question regarding the extent of structure, especially in liquid condensates. You know, we've become really quite good, first with crystallography, then solid state NMR, and then cryo-EM in determining the structure of insoluble aggregates and amyloids. But it's really still quite tough to determine the level of structure inside liquid condensates. And I'm not going to answer this question here, but at least I think that the mutagenesis will provide a very good beginning to start answering this. So we made a very simple assumption that is the two residues that are close in the three-dimensional space would be more likely to give rise to genetic interactions. So if you see here, if you mutate A and C in this slide, they will be very likely to just give an additive effect. So the toxicity of A plus the toxicity of C, sorry. But if you mutate A and B, they will be much more likely to be epistatic and give rise to an unexpected outcome. And so we could quantify this unexpected outcome and literally build contact maps where we highlight the most interacting residues, the most interacting residue pairs. But what we put in the contact map is actually more of an integrated measurement where we also take into account not only if you and I are interacting, but also how we both interact with the rest of the people in this room, which increases the specificity of our measurement. We can scan then these contact maps for secondary structure patterns, where residues that are 3 or 4 apart in an alpha helix would be more likely to interact, or just 2 apart in a beta strand would be more likely to interact, and we score them for significance. And the 2 most significant peaks for secondary structure inside this putatively disordered domain were actually a peak for a specific type of beta strand, which is this aromatic rich kinked beta strand, and then a peak for a alpha helix, which are both taking place inside the hotspot of toxicity, actually. And basically from epistasis measurement, what we have is a signature of their presence in vivo. This is the first evidence that the TDP prion-like domain may actually be partially structured inside the cell. But there's in vitro evidence for these structures, which was quite comforting for us. The first one comes from X-ray of peptides from the prion domains from the Eisenberg lab. And the second one comes from in solution NMR, actually from Nicholas Fauzi's lab. So, and the key thing about these two elements is that from in vitro studies, we know that they're both highly stabilized by self-interaction of TDP monomer with other TDP monomers. So they do exist in an aggregated state or an assembled state, at least. A conservative model of how TDP self-assemble would see, obviously they're not compatible, these two secondary structure elements. So you would see the helix forming first in the process of aggregation and then maybe evolving to a beta strand. This is obviously speculation. I'm not saying, I will clarify what I'm not saying and what I'm saying, I think, right now. So what I'm not saying is that we know that these structures are particularly good or bad for the cells. We don't know that. We know that mutations that are stabilizing or destabilizing this structure have unexpected outcome on toxicity. But we're doing more high resolution sort of work to actually be able to say what they mean in the onset of the cellular phenotype. What I am saying is that they do form in vivo, which is pretty striking because it means that also deep mutagenesis is a strategy that may allow us to determine if these domains that we think are disordered are actually disordered inside the cell or are maybe partially ordered thanks to interaction or aggregation. In this specific story, we realized that the cytoplasmic aggregation of TDP-43 is actually not toxic, but that toxicity may arise from liquid condensates at the nuclear periphery. And I will actually move on now to talk shortly about something else we're currently doing in the lab and which I think may be of interest for some of you here. So I think that, before, let me just mention that actually in this work, we also developed a pipeline to sort of standardize the way we analyze this data in the lab. And if you are happy to test it, the manuscript is under revision, but if you're happy to test this or play around with it, please note down this link. Something that was striking for me during this work was that really we could explain such a huge amount of protein, of the toxicity of such a huge number of sequences on the basis of just one property. And we decided to just test this on different data sets published around. And one of the data sets that we took was actually generated by Doug's lab. And what they did in this case was to take the amyloid beta peptide, which, for those of you who don't know it, aggregates extracellularly in Alzheimer's disease. And they fused this to the DHFR, which is a very nice way to track the solubility of the peptide. If the peptide aggregates, so does the DHFR, and you're not able, well, the yeast cell is not able to survive in the presence of methotrexate. And the opposite once the peptide is soluble. And so they could map, let's forget for a moment about like liquid condensates and so on. This is a very efficient hydrophobic aggregation-prone peptide that aggregates extracellularly, by the way. So what they could map is solubility, and that's what you see here with red hinting at the very soluble stuff and blue at the non-soluble things. And as you may expect here, again, hydrophobic amino acids are killing solubility, and the opposite is true for polar and charged amino acids. So we could see this effect of amino acid type again on a different system where a different selection method was used. And so we used, again, our PC1 hydrophobicity to try and predict this data. And well, not surprisingly, it predicts exactly the variance in solubility to the same extent with which we could predict the variance in toxicity for TDP-43, suggesting that we may be after actually a similar type of hydrophobicity-driven mechanism of aggregation inside the yeast cell. At the same time, well, we tested a few other predictors here as well, predictors of aggregation or of intrinsic disorder, but hydrophobicity by itself was the one property able to explain the data the best. And at the same time in the lab, I had a student working on some other reporter of aggregation that we wanted to use for a bunch of prion domains, for example. And this reporter is actually based on a fusion of the sequence you want to the sub-35 nucleation domain. Sub-35 is a classical yeast prion, and you can fuse just the nucleation domain of the prion to any protein of interest. And in this case, what happens is that if your protein or peptide is aggregating, it will also sequester the endogenous sub-35, which will stop functioning. And its job would be, in this case, to recognize a stop codon in the ADE gene. So this means that if sub-35 doesn't do this, you will have a read-through of translation, and you can actually grow in the absence of adenine. So basically what happens in our selection assay is that you sequence in non-selective conditions after expressing the peptide, and then we plate on millions of plates without adenine, and we sequence again. And the ability to grow without adenine is therefore a proxy, let's say, of aggregation propensity. And the map we get is actually a bit different with a very strong, so here you see in red, again, solubilizing mutations, and in green, aggregation, let's say mutations leading to increased aggregation propensity. And we have definitely a N-term to C-term effect. This is fairly new data, so I'm just gonna say a few quick things about it and what I think it means. We have a massive N-term to C-term effect with actually mutations having more of an impact and becoming more and more solubilizing, going towards the hydrophobic C-term of the peptide. And as you expect, well, you don't really, well, not as you expect, but you don't really see an effect of amino acid type that much here, okay? So we don't have this clustering that was so evident in our previous TDP data set and in the A-beta data set that I just, from Doug's lab. So as one can expect, once we try to explain this data with hydrophobicity, we perform really poorly. So I think it's very interesting because it will show you that actually just by changing slightly the reporter of aggregation and your selection system, you may end up being after a different mechanism, but in this case is not explained by hydrophobicity, but in the other A-beta data set that I showed you was actually explained really well by hydrophobicity. This is very much speculation, but what I think this may mean is that actually in yeast you can have two different types or maybe more of aggregation mechanisms. One that is really a bit aspecific and hydrophobicity driven, and it's what we've seen in TDP-43 or what we can see with the DHFR fusion, and one that is possibly a bit more specific and really amyloid-like, and is what we see with this other type of prion fusion. I can't be sure about this yet, but what I was happy to see is that if you are familiar with Alzheimer's mutations, you know that the aspartic and the glutamic in position 22, 23 are key spots for familiar forms of the disease. And indeed, all the mutations that we see here are all extremely aggregation prone in our assay. And if we test all the 10 officially reported familiar Alzheimer's mutations, these are all associated to very early onset and aggressive forms of the disease, we actually can discriminate them with a strong significance. So we may be after a mechanism that is actually relevant to disease. So I will just say that the fusion to the sub-35 nucleation domain provides an alternative way to track aggregation, where selection is not based merely on hydrophobicity, and that this assay seems to significantly discriminate familiar Alzheimer's disease variants. And with this, I will conclude actually by thanking, well, the TDP work is work that I started at the end of my postdoc at the CRG. So I'll have to thank especially Ben Lehner's lab for everything that came from it. It's work that I shared with Andre, who is also responsible for the pipeline that I mentioned. And then Mireia is my PhD student who is involved in the beta project currently. And thank you for your attention.